Good morning, everyone. It's a pleasure to see so many future pharmacists and biologists here today. We are about to dive into what I consider the most real-world aspect of pharmaceutical sciences. You've spent years in labs learning how to make molecules bind to receptors. You've mastered chemical synthesis and molecular docking. But today, we ask the much harder, much more expensive question: can that molecule survive the human body, and more importantly, can the human body survive that molecule? We are discussing ADMET, the 5 pillars that determine whether a brilliant lab discovery actually becomes a life-saving medicine or remains just a footnote in a dusty research journal. This is where high-flying theory meets the grounded reality of human biology. Let's start with some sobering statistics that every student in this room should memorize. In the world of drug discovery, we are essentially looking for a needle in a haystack, but the haystack is 1,000 miles wide and occasionally on fire. For every 10,000 compounds we screen at the start of a project, only one will ever reach the pharmacy shelf. 1 in 10,000. Now, why do the other 9,999 fail? Historically, we thought drugs failed because they didn't work, what we call lack of efficacy. But the data tells a different story. Throughout the 90s and early 2000s, the leading causes of failure were actually poor pharmacokinetics and unexpected toxicity, ADMET. Our goal in the preclinical stage is to act as the body's intelligence agency. We want to predict human response and identify lethal hazards before a single human volunteer ever takes a dose. This data is what allows us to calculate the first-in-human safe starting dose. We aren't just researchers, we are the gatekeepers of patient safety. Before a drug touches a human in phase 1 trials, it has to survive this rigorous preclinical gauntlet. Think of it as an elite military training program for molecules. First, we have target validation. We spend months ensuring the lock we are trying to pick is actually the biological cause of the disease. If you pick the wrong lock, the best key in the world is useless. Then, lead identification. We use high-throughput screening to find any chemical that fits that lock. But look at the third step, lead optimization. This is where chemistry meets ADMET. We don't just want a molecule that binds, we want one that is drug-like. It needs to be stable, soluble, and non-reactive. Finally, we reach the safety assessment. Here, we use in vitro and in vivo models to establish safety margins and find the NOAEL, the no observed adverse effect level. This entire mountain of data is what we submit in an IND, investigational new drug application. Without an approved IND, the project stops dead. No IND, no clinical trials, no medicine. It's that simple. Let's break down the acronym that will define your careers. ADMET. Absorption is about the entry. How does the drug get from the outside world into the blood? Distribution is the map. Once it's in the blood, where does it settle? Does it go to the brain? The fat? Or does it just float in the plasma? Metabolism is the modification. This is how the body, primarily the liver, tries to chemically dismantle the drug because it sees it as a foreign invader. Excretion is the exit. How do we finally get rid of the drug? And finally, toxicity is the consequence, the adverse effects that happen at any step. Together, the first 4, ADME, define the pharmacokinetics, or what the body does to the drug. The last one, toxicity, defines the safety margin. If you don't understand these 5 factors, you don't understand your drug. You just have a chemical, not a medicine. Absorption is the movement of the drug from its site of administration into the bloodstream. It is the drug's first major hurdle. Chemically, it's a battle of properties. We look at lipophilicity, your log p-values, molecular weight, and solubility. If you take a drug orally, which is what every patient wants, it has to survive the acid bath of the stomach and then permeate the intestinal wall, which is a major biological barrier. We talk about bioavailability, the percentage of the dose that actually reaches the systemic circulation unchanged. Now, if you give a drug via 4, bioavailability is 100% because you're putting it directly in the blood. But for a pill? If a drug is too hydrophilic, it's like trying to push a magnet through a wall of oil. It just won't cross the lipid membranes of the gut. Conversely, if it's too lipophilic, it might get stuck in the fat layers and never reach the blood. It's a delicate balancing act. Once the drug successfully reaches the blood, it doesn't just sit there waiting for instructions, it moves. This is distribution. We use a theoretical term called volume of distribution, VD. If a drug has a high VD, it's not in the blood, it's sequestered in tissues like fat or muscle. This is common with highly lipophilic drugs. If it has a low VD, it's likely bound to plasma proteins like albumin and stays in the circulation. And we cannot talk about distribution without mentioning the blood-brain barrier, BBB. This is the most restrictive border in the human body. The endothelial cells in the brain are stitched together with tight junctions that act like a high-security fence. If you are designing a drug for Alzheimer's or depression, it must be small and lipophilic to cross that fence. However, if you're designing a drug for a skin rash or hay fever, you might actually want it to not cross the BBB to avoid making the patient drowsy. Distribution isn't just about where the drug goes, it's about where it shouldn't go. Next, we come to metabolism, primarily in the liver. Think of the liver as the body's high-tech reprocessing plant. Its primary goal is to take lipophilic drugs and make them more hydrophilic, water-soluble, so the kidneys can flush them out. Phase 1 usually involves the cytochrome P450, CYP450, enzyme family. These enzymes are the workhorses of the liver. They oxidize, reduce, or hydrolyze the drug to expose a polar handle. Phase 2 then tags that handle with large, polar endogenous molecules like glucuronic acid or sulfate. A critical warning: the first pass effect. When you swallow a pill, it goes from the gut to the liver via the portal vein before it reaches the rest of the body. Some drugs are so heavily metabolized on this first pass that only 1% makes it to the heart. We also must obsess over drug-drug interactions, DDI. If a patient is taking drug A, which inhibits a CYP enzyme, and you give them drug B, which is cleared by that same enzyme, drug B will build up to toxic levels. This is why we tell patients not to drink grapefruit juice with certain statins. Grapefruit is a potent CYP inhibitor. Finally, we have to get the drug out. No drug should stay in the body forever. This is excretion. The kidneys are the stars here. They filter the blood, they secrete waste, and they reabsorb what's useful. If a drug is polar and water-soluble, it's going out in the urine. This is why we worked so hard in the metabolism stage to make it polar. But don't forget the biliary route. Some large or highly polar molecules are excreted by the liver into the bile and eventually leave the body via the feces. As clinicians and researchers, you must always consider the patient's organ health. If a patient has renal or kidney or hepatic or liver impairment, the drug stays in the body longer, the half-life increases, and the risk of toxicity skyrockets. In this field, dosing is never one size fits all. It's a calculation based on how well the body can say goodbye to the molecule. Now we turn to the most critical, T in ADMET. Paracelsus famously said 500 years ago, "The dose makes the poison." Everything, even water, even oxygen, is toxic at a high enough dose. In safety assessment, we look at the therapeutic index. This is the ratio between the toxic dose, TD50, and the effective dose, ED50. Think of it as your margin for error. A drug like Valium has a wide therapeutic index. It's very safe. A drug like digoxin for the heart or lithium for bipolar disorder has a narrow therapeutic window. With these drugs, a tiny mistake in dosing or a slight change in the patient's metabolism can be fatal. We also distinguish between acute toxicity, effects that appear immediately after one large dose, and chronic toxicity, which is the slow buildup of damage over weeks or months, like the liver damage you see from chronic alcohol or acetaminophen use. Before we ever consider using animal models, we screen for showstoppers, using in vitro tests in the lab. First, the Ames test. This uses a special strain of salmonella to see if your drug causes mutations in DNA. If it's positive, the drug is likely a carcinogen. In 99% of cases, a positive Ames test kills the project immediately. Then there is the hERG assay. This is a fascinating and terrifying test. It checks if a drug interferes with the hERG potassium channel, which manages the heart's electrical recharging phase. If you block this, you cause QT prolongation, which can lead to sudden, fatal cardiac arrest. This test alone has ended thousands of drug programs. And finally, cytotoxicity assays. We dump high concentrations of the drug on human liver cells, hepatocytes, and watch for cell death. We want to find these red flags now, while the drug is still in a vial, not later when it's in a patient. If a drug passes the big three in vitro tests, we move to in vivo animal models. This is not optional. It is a legal and ethical requirement before human testing. We typically use two species, a rodent, the rat, and a non-rodent, usually a dog or a non-human primate. We perform single-dose studies to determine the maximum tolerated dose, MTD. Then we do repeat dose studies, where animals are given the drug daily for up to 6 months to see what happens to their organs over time. We also check for reproductive toxicity. We must know if the drug affects fertility or causes birth defects in a developing fetus. The key here is the balance between pharmacodynamics, PD, what we want the drug to do, and toxicodynamic, TD, the unintended damage it causes. A blood pressure drug that successfully lowers BP/PD but causes severe liver inflammation TD is a failure. We are looking for that perfect, elusive balance. If a drug passes the big three in vitro tests, we move to in vivo animal models. This is not optional. It is a legal and ethical requirement before human testing. We typically use two species, a rodent, the rat, and a non-rodent, usually a dog or a non-human primate. We perform single-dose studies to determine the maximum tolerated dose, MTD. Then we do repeat dose studies, where animals are given the drug daily for up to 6 months to see what happens to their organs over time. We also check for reproductive toxicity. We must know if the drug affects fertility or causes birth defects in a developing fetus. The key here is the balance between pharmacodynamics, PD, what we want the drug to do, and toxicodynamic, TD, the unintended damage it causes. A blood pressure drug that successfully lowers BP/PD but causes severe liver inflammation TD is a failure. We are looking for that perfect, elusive balance.